Hello everyone. We are going to start lecture on the placenta. So you all know that the uteroplacental circulation began much early when the trophoblast differentiated into two layers, the syncytial trophoblast and the cytotrophoblast. And when the syncytial trophoblast started to burrow into the uterine endometrium, creating lacunae, and the maternal blood started to flow in those lacunae, the spaces. As early as that, during the end of the second week, the uteroplacental circulation began. So that was the first contact between the maternal and the fetal part. So now we will see the full development of the placenta. It is an organ that connects the developing fetus to the uterine wall. This placenta is called as a fetomaternal organ, and hence it has got two parts, fetal part and the placental or the maternal part. So this organ allows nutrients uptake, waste elimination, and gas exchange via the mother's blood supply. Let us see the next slide. As I said to you, it functions as a fetomaternal organ having two components. The fetal part enclosing the embryo and all of its other membranes that develops from the chorionic sac. If you remember in the early part of the development how the chorionic sac developed, right? The chorion, which is nothing but the two layers of the trophoblast and the extra embryonic mesoderm, somatoporic mesoderm. Okay, the maternal part is that part that is derived from the endometrium, which is called as decidua basalis. The area of endometrium between the implanted chorionic vesicle and the myometrium, which becomes the maternal part of the placenta. Decidua. Decidua means to shed. So if there is no fertilization, then it is this part. That's why the endometrium gets shed during the menstruation. So decidua means to shed. So the part of the endometrium that undergoes the reaction, the glands increase and they become vacuolated, become filled with glycogen and fat. Now this is called as decidua reaction, and the endometrium divides into decidua basalis, parietalis, and capsularis. In the later part of the slides, we are going to discuss on this. So just to bring you that what it is the endometrial reaction that undergoes changes as soon as there is implantation. So this is decidua basalis, which takes part in the maternal part of the placenta. And as I said you, placenta helps in protection, nutrition, respiration, excretion, and also hormone production. So as you could see in this diagram here, as I had told you just a few while ago, that as early as now it is 5 to 6 days, despite the end of the second week, almost there is a connection, there is a uteroplacental circulation has started to develop. You can see here by the end of the 5th and 6th day, the syncytial trophoblast, which is the layer of the trophoblast, starts burrowing into the uterus. You can see here, this is the endometrium of the uterus, and this is the start burrowing in the uterus. In the next slide, you can see that there is early formation of trophoblastic lacuna, which I just now explained to you. And as this lacuna gets bigger and bigger, it burrows into the maternal endometrium, and the maternal blood vessel starts flowing its blood into it, so establishing the uteroplacental circulation. Okay, so what you see in this diagram, if you remember your earlier lectures, you can see here there is a layer of cytotrophoblast, right? And this is syncytiotrophoblast, which is multinucleated, no cell line, no cell boundary. Okay, so that it has a capacity to burrow. Okay, it has highly mitotic structure, and you have the amniotic cavity is seen here, and this is the blastocyst cavity which is forming the primary yolk sac. Let us move on from here. As you could see here now, primary villi are started to form. If we can recollect primary villi, secondary and tertiary villi, let us recollect those lectures where the villi formation. So early in the third week, mesenchyme grows into the primary villi, forming the core of the mesenchymal tissue. Thus, the secondary chorionic villi are formed over the entire surface of the chorionic sac. So, what are primary villi? When the cytotrophoblast goes into the syncytiotrophoblast, now this is two layer. Outer is syncytiotrophoblast, inner is, you can see here, inner is cytotrophoblast. This is primary villi. So, in the early third week, the mesenchymal core starts, there it is, it starts going into the, so it starts growing into the primary villi, forming a core of the caval tissue. Thus, the secondary chorionic villi are formed over the entire surface of the chorionic sac. Then some mesenchymal cells in the secondary villi differentiate into blood vessels and capillaries, forming the tertiary chorionic villi. And the capillary in the villi fuse to form the arteriocapillary network. So this is how primary, secondary, and tertiary villi are formed. This shows you the primary villi. Here you can see the secondary villi forming as the first layer is syncytioblast, the second is cytotrophoblast, and third is the mesenchyme invading into it, right? And then you have the formation of the. Now let us orient here to this diagram. You can see on your side, this is the maternal side or the uterine side. This is the maternal side. Okay, and this part is the fetal side. Clear? Right. Then what you can see here in this diagram, just orient yourself. Now, we are here, uterine tissue now, which is called as decidua basalis. You can see the primary villi has been formed. This purple is the syncytiotrophoblast. Can you see this? This is syncytiotrophoblast that has already burrowed into the uterine endometrium, in which is now the layer of cytotrophoblast burrowing. So that forms the primary villi. Then the third one here is the mesenchyme getting into the cytotrophoblast, forms secondary villi. And as the blood vessels start developing here, it forms the tertiary villi. So now the villi is complete. Formation of the chorionic villi is complete. Can you see that? We started from the primary villi to secondary villi to tertiary villi, and now you can see they are forming something called as anchoring villi. Okay, here, as you can see here, the cytotrophoblast cells, they reach all the till the uterine endometrium. Okay, all the way here, and they create now called as cytotropoblastic shell. Is it clear? So the cytotrophoblast, which were earlier just below the syncytiotrophoblast, now they have gone beyond it, and they have created a shell called as cytotropoblastic shell. So you can see anchoring villi are anchored between the chorionic plate and the cytotropoblastic shell. Is it clear? They are anchored. That's why the word anchoring. And you can see it is made up of truncus villi. This is the truncus villi. These are the branches of the villi, the free villi, which are in between the space, now called as intervillous space. These are the floating villi. Can you see those? And this is the truncus villi, the main trunk. And they are in a space called as intervillous space. This is intervillous space. I will just label for you here. I'll write in short form, intervillous space. What flows in the intervillous space is the maternal blood. Now, is it clear, this diagram for you all? Okay, let us on to the next slide. You can see here, secondary villus becomes tertiary, and when the blood vessels penetrate its mesenchymal core and newly formed branches. So this occurs towards the end of the 3rd week of pregnancy. See the timeline. It occurs toward the 3rd week of pregnancy. We started here and now we are here somewhere. So by there, everything is clear, the 3rd week of pregnancy. Now, the terminal portion of the villus consists of a cytotrophoblastic column and a thin sensitive trophoblastic layer, which is bedded in the maternal blood. A further development of the villus tip occurs when, under hypoxic condition, the cytotrophoblastic column expands distally, penetrating the sensitive trophoblast. Now, just orient yourself to this diagram so that you now know I'll just go to the whiteboard. Okay. Now, we said here is the chorionic plate. In the blue, this is the maternal side. This is the maternal side. And now this is the fetal side. So, what did we say? First is the syncytial trophoblast in the red. Okay. Then is the cytotrophoblast, and then is the mesenchymal core, and then you have the in the yellow the blood. So now the cytotrophoblastic core goes beyond the syncytial trophoblast. Now this forms the cytotrophoblastic shell. Are you getting it? So now you have those villi. Just let me draw for you the villi. It will be much clearer for you. I'm drawing in the red the whole thing. Are you getting it? There it is. Are you getting it? This is called as this is one villi, this is another villi. Now this is intervillous space. Now from here, the maternal blood will go inside and it will bathe the villi. Understood? So this is called as anchoring villi. Anchoring villi. It anchors between the chorionic plate and the maternal side. And now you know what is cytotrophoblastic shell. Clear? Okay. Let us go back to our slide where we left. Okay. Here it is. So this is what I was about to tell you. There I am, the cytotropoplastic shell column adjacent to the maternal decidual cells and spread over them surrounding the embryo, right? Anchoring villi represent the real attachment point between the maternal and the embryo tissue. Is that clear now? This is the cytotropoplastic shell you can see in the green, right? And the cytotropoplastic shell forms the column here and goes beyond the syncytiotropoblast, which is purple in color here in the diagram, and the green is all cytotropoblast, so it forms a shell, right? Okay, moving on to the next. Unattached branches, which are called as floating villus, you saw those dangling freely in the maternal blood that fills the space between the chorionic plate and the outer cytotropoplastic shell, which is the intervillus space. Done? Right. And now all the surfaces of the villi, the chorionic plate, the cytotropoplastic shell are in contact with the maternal blood, are lined with a continuous layer of, you can see in the diagram, they're continuously lined by the syncytiotropoblast. Clear? Okay. Villi and the outer surface of the chorionic plate are bathed in the maternal blood. You already I told. That is why the human placenta is hemochorial type. It is a hemochorial type. There are various types of placenta in other animals, but in human you have hemochorial types because the maternal blood bathes the outer surface of the chorionic plate also. Right. So now in this diagram, just orient yourself. Embryo, which is attached by a body stalk, now called as umbilical cord, is suspended in the chorionic cavity, which is bounded by the chorionic plate. Are you all orienting yourself? You can see here, this is the umbilical cord, which has been marked with an arrow, and you can see the villi, the spiral artery, which is taking the maternal blood in the intervillus space. All that white space between the villi is the intervillus space. Yes, look at here. This is outer cytotropoplastic shell, the green one, and this is the chorionic plate, which is nothing but extraembryonic mesoderm. Clear? And here is the chorionic cavity, and this part is the decidua capsularis, which does not take part in any function of forming placenta. It is all over here, which is decidua basalis, that forms part in forming the maternal part of the placenta. So you can see here the same thing which we explained in reference to this diagram. You can see the chorionic villi extend outward from the chorionic plate, and the tropoplastic covering is in continuous with the chorionic plate. And the chorionic plate consists of extraembryonic mesoderm made with the tropoblast. Right. So, what is about the umbilical cord? So the umbilical cord comes from the fetus attached close to the center of the placenta. It is containing two arteries and one vein. They branch on the fetal surface to form the chorionic vessels, and these are umbilical arteries. Now, in our body, there is an exception with regards to two arteries, blood vessels. One is the pulmonary artery that carries the deoxygenated blood to the lung, and another is the umbilical artery that carries deoxygenated blood, right? So two arteries are the exception in our body which carry deoxygenated blood. And it supplies blood to the uterus, the uterine artery, and the blood in the umbilical vein is oxygenated by the spiral arteries in the placenta. So the most veins, as I had said you about this exception, most veins carry deoxygenated blood from the tissue back to the heart, exceptions being the pulmonary and the umbilical vein, which both of which carry oxygenated blood. So how does the spiral artery invade the following implantation of the blastocyst? The trophoblasts continue to invade the endometrium until they reach the spiral artery. Then the trophoblast begins to remodel the spiral artery by replacing its smooth muscle and endothelial cells. Increases the blood flow, which is essential for the embryo. Lack of spiral artery modification can lead to complications such as preeclampsia and intrauterine growth retardation. Okay, so these are the modification of the spiral artery. Leading to increase in the blood flow, and if this modification does not take place, it will lead to preeclampsia and intrauterine growth retardation because of lack of supply. Preeclampsia is a pregnancy complication characterized by high blood pressure and signs of damage to other organ systems like liver and kidney. Usually it begins after the 28 weeks of pregnancy in women whose blood pressure has been normal. So this is preeclampsia for you. What is the main purpose of establishing uteroplacental circulation is to bring food and oxygen and to remove the waste from the embryo. So the establishment of uteroplacental circulation is accomplished by erosion of the wall of the spiral artery of the uterus and modification of the spiral artery so that they can provide increasing flow of blood at a very low pressure to bathe the syncytial trophoblast surface of the placenta. So this is the structure of the placenta here. Just orient yourself to the structure here. You have the chorionic plate. So this is the fetal side where the umbilical cord is attached. And this part is the maternal side. You can see the spiral artery taking the blood. First it goes like a high pressure and then falls like a fountain to bathe all the villi in the intervillous space. After bathing the villi, the blood is drained via the endometrium veins. The specialized invasive cytotrophoblasts, they migrate from the anchoring villi to invade spiral artery and then displace the cells of the spiral artery. So they become wider. Now, I had told you about this changes in the uterine endometrium within few days after the implantation. Stromal cells of the endometrium undergo a decidual reaction, which I mentioned you in the beginning of the chapter, to form decidual cells. Now, three parts are formed: decidua capsularis, decidua basalis, and decidua paritalis. Let us see. As you can see here, in this diagram, the decidua basalis forms the maternal part of the placenta, and it is this part which is covering. The decidua capsularis covers the conceptus, you can see here, and the decidua paritalis is the rest of the endometrium. Okay, so the three parts. As you can see here, the endometrium lining of the uterus, known as decidua, consists of three regions named by location. Decidua basalis is between the blastocyst and the myometrium, to be more precise. Capsularis, that covers the implanted blastocyst, as I said you, that covers the conceptus, and decidua paritalis is all remaining of the endometrium. And why does this happen? It happens because the stromal cells in the endometrium, the glands, they increase, they become vacuolated and with lipid, and that causes this reaction to take place. So in human, what is chorion? Chorion, as I said you in the beginning again, is a layer that consists of trophoblast, both the layers of trophoblast, and the extraembryonic mesoderm. So chorion forms a complete covering, chorionic vesicle that surrounds the embryo, the amnion, the yolk sac, and the body stalk, which is the umbilical. So as we know, placenta is formed of two components, fetal and the maternal component. Let us see the difference. The fetal component consists of a wall of the chorionic plate and the chorionic villi, as you saw in the diagram, and the maternal plate is represented by decidua basalis, which is also covered by outer cytotrophoblastic shell, and there is an intervillous space between the fetal and maternal components of the placenta, which is occupied by the maternal plate. So a typical placenta weighs around 500 grams. The fetal side of it is shiny because amniotic vein is present over there, and the fetal side you have attachment of the umbilical cord. With umbilical vein and artery. As you can see here, this is the picture of a placenta where you can see the structure of the placenta is lobulated. Now this is the maternal side. There is no umbilical cord attached here, so this is not going to be a fetal side. This is the maternal side. This is a full-term placenta. What you see, those lobulated structures are now called as cotyledons. What are cotyledons? About 15 to 20 slightly bulging villus areas, and their surface are covered by the shreds of the decidua basalis from the uterine wall. So these are formed by these structures are called as cotyledons. Okay. So we will further. So the maternal side, as you can see here, 35 lobes. Placental septa between the lobes which arise from the decidua basalis and extends towards the basal plate. Within a placental lobe, several cotyledons, each consisting of main stem villi and all its branches. If we go back to our diagram here, it's very clearly mentioned for you. I will just mention in the diagram so that it becomes very easy for you to know the placental septas. So can you see those septas being formed here? Okay. These are the cytotrophoblastic shell columns, and that further will form the septas, and that will give rise to those lobules, which are now called as cotyledons. Here, they are between the lobes which arise from the decidua basalis and extends towards the basal plate. So several cotyledons, each consist of main stem villus and all its branches. So at the delivery, you have first the pregnant lady will have first contraction for the baby to come out, and after 5-10 minutes there is a second contraction for the placenta to come out. So when the placenta comes out, and it is our duty as a physician and whoever is present at the delivery time to check the surface of the maternal for all the cotyledons to be intact. Okay, if any of the cotyledons are missing, that can give rise to postpartum hemorrhage. So always we have to inspect the placenta for any missing cotyledons, because as you can see here, it consists of main stem villi and all its branches. Okay, so we have to be careful at the time of the delivery. So incomplete partition between the placental cotyledons are covered with trophoblast and contain a core of maternal tissue. So Villus core consists of blood vessels and mesenchyme, and you have half Bauer cells, which are like fetal macrophages, which are scattered among these mesenchymal cells, right? And this villus core is covered by a continuous layer of syncytial trophoblast with minimum number of cytotrophoblasts beneath it. The syncytial trophoblast surface is covered by microvilli, which greatly increases the surface area of the placenta for transport of material, hormones, growth factors, enzymes, and proteins. So any change with the microvilli size or the density, if it is not constant, like change with increasing placenta age and differing environmental conditions. For example, poor maternal nutrition or oxygen transport, microvilli they increase. Poor adaptation to adverse condition can lead to newborns with low birth weight. So certain conditions, increasing age of the mother, okay, with different environmental condition can affect the villi, and then that can affect the development of the placenta, resulting into low birth weight. So with this, we come to an end of this chapter. Hope you understood it. Thank you.